Alright, welcome back for the last video in this particular set of lectures. So we've had a good long set of discussions on proteins, what proteins are made of, amino acid classes, how proteins folded. In this video, we're going to talk about different classes of proteins and what proteins tend to do. Now, this is going to be something that we touch on and continue to touch on as we go throughout the semester, but I think I would be remiss if we didn't at least have a little bit of a discussion on this now. Now, for different classes of proteins, we are going to use specific proteins as examples, but I do not want you to sweat the details of those proteins too terribly much. So just be aware that when you start talking about a particular type of protein, practically speaking, you're going to be studying that particular type of protein. So you're going to want to eventually know as much about that protein as humanly possible. So just bear that in mind. Okay, so this diagram, which comes from a general biology textbook I used to teach out of, does a pretty good job of summing up different classes of proteins. So you've got channel proteins, carrier proteins, cell recognition proteins, receptor proteins, enzymes, and junction proteins. Now, this diagram actually specifically shows just membrane proteins, or proteins that are inserted or some way anchored to the cell membrane. But even so, this does a pretty good job of summarizing what most proteins tend to do. So we'll talk about examples of each as we go along here. So I have here a little bit of a description of each type that we saw in the previous diagram. Enzymes are almost always going to be proteins, as we mentioned. A good example of an exception to that rule is the ribosome, as the peptidyltransferase enzyme activity actually belonged to the ribosomal RNA and not the protein part of the ribosome. But in spite of that, almost every enzyme is going to be a protein. So we will have a whole chapter and lecture dedicated to learning about enzymes, so we're not going to talk about it too much right now. But the idea with enzymes is that enzymes will speed up the rates of chemical reactions that would otherwise occur far too slowly to be useful to cells or biological applications. Enzymes can either be soluble, meaning they're just floating around in bodily fluids, or they can be bound to the cell membrane, as we saw in the previous slide. Secondly, we've got transport proteins. These proteins are going to facilitate the movement of molecules either throughout the cell or more commonly through the cell membrane. Now, if this was my human physiology class, I would probably spend a whole chapter talking to you about how the cell membrane is semi-permeable. Some molecules can move through the membrane, but most molecules cannot. So transport proteins are going to basically allow molecules that would otherwise not be able to get through the membrane to be able to do so. A good example of solutes that cannot move through the membrane ordinarily are sodium and potassium. So a specific type of protein, a transport protein that we're going to look at called the sodium-potassium pump, is going to move sodium and potassium ions across the membrane for the sake of maintaining physiological homeostasis. Cell recognition proteins are almost always going to be found on the outer surface of the cell membrane, and they basically give the cell a particular fingerprint that can be recognized by other cells in the vicinity, usually the immune system. So the example we're going to look at here are the blood-typing antigens A and B. These are actually proteins that are found on the surface of red blood cells. Junction proteins are proteins that give cells structural integrity, give them strength and resilience, and then they'll also perform important physiological functions like sealing the spaces between epithelial cells in the intestines so bacteria and other creepy-crawly organisms cannot move from the food that we eat and into our bloodstream. That would be very bad. And then also different types of junction proteins can allow excitatory signals to spread throughout heart tissue, which is important for allowing our hearts to beat. And then finally, receptor proteins are proteins that bind to chemical signals and transduce those signals into physiological responses. So for each type of protein, we are going to give a particular type of example. And like I said in the intro to this video, don't sweat the details of each example too much, but just be aware that when you study a protein, like we are going to be studying tyrosinase this semester in lab, which is an enzyme, you're going to want to do everything in your power to learn as much about that particular protein as possible. So just think of these examples as kind of a roadmap for where you would want to get to go for a protein that you are studying. Okay, so first, just a little intro on enzymes. So an enzyme, again, is a catalyst that speeds up the rate of a chemical reaction. Each metabolic reaction will usually have its own unique enzyme, again, most of which are going to be proteins. So a particular metabolic reaction, usually it's going to be far too slow in the absence of an enzyme. So a lot of our proteins in our body are enzymes that get our chemical reactions to go inside and outside of our cells. Each enzyme is going to have a special binding pocket, again an exemplification of how important shape is, called an active site. The active site is where the substrate molecules go, the substrates being the molecules that the enzyme works on. So the active site is going to be shaped in such a way that only that particular substrate can bind there and other substrates cannot. And this is going to ensure that a particular enzyme really can only catalyze one particular type of chemical reaction. And again, substrate is the special name we give to reactants that interact with enzymes. And then another important note is that the active site will contain the side chains of very important amino acid side chains. We call these catalytic side chains. These side chains are going to actually actively participate in the chemical reaction by forming and breaking chemical bonds in the substrates to produce whatever the product is meant to be. The amino acid side chains of the enzyme actually get the chemical reaction to go by forming and breaking bonds. Okay, let's talk about an example of transport proteins. Now, this is probably what you might think is kind of a complicated and involved example, but I promise we're not going to look at it too terribly deeply. So this is what happens when you eat food. So you eat food, let's just say you're eating a bagel, let's say a bagel. You eat a bagel, you chew the bagel goes down to your stomach where you continue to break down that bagel into individual sugar molecules, monosaccharides. Those monosaccharides are then going to move into your small intestine where you are going to try to absorb that sugar across the cells that make up the wall of the intestine and into the bloodstream. You've got to get those nutrients into the bloodstream so that you can circulate and deliver those nutrients to all the cells throughout the body that need those nutrients, right? So what we're trying to accomplish here is we want to get this glucose, which is inside the hollow portion of the intestine here where our food is, we want it to move across the top part of this cell membrane and then across the bottom part of this cell membrane. But the problem here is all the solutes that you see here, including glucose, sodium, and potassium, these are the types of solutes or molecules that cannot move across the membrane unless they have a transport protein that is willing to let them across. So in this particular example, we've actually got 3 different transport proteins that work together to allow us to absorb glucose into our bloodstream from our intestines. You've got this first transporter, which is going to move glucose and sodium inside the cell at the same time. You've got a second transporter that is going to move glucose into the bloodstream, and then you've got the sodium-potassium pump down here at the bottom that is going to function to make sure that we always have a healthy supply of sodium up here to allow glucose in. Okay, so for our example of cell recognition proteins, we're going to use blood typing as our specific example here. Again, we're not going to go too crazy deep into this, but you are probably peripherally aware of the blood typing system. There's 4 major blood types, A, B, AB, and O. So what your blood type is determined by which types of proteins, which types of cell recognition proteins you have on the surface of your red blood cells. There are only 2 types of these proteins. They are called A and B. If you've only got A antigens, then you're type A. If you've got B antigens, then you're type B. If you've got both of them, you're type AB. And if you've got neither of them, you are type O. So basically, the idea here is that if you are type A, you've got these A antigen cell surface proteins here, and in your blood plasma, you've got antibodies against the B antigen, against the antigen you do not have. So if a red blood cell were to make its way into your body that is type B, those antibodies are going to recognize that antigen and be like, okay, you're not supposed to be here. Our antigens are supposed to be type A. So this is the sort of thing that we're talking about with cell recognition. Having these types of proteins on the surface of cells allows the body to recognize what is native to the body and what is not. So this is obviously very useful for fending off infections from microorganisms, and then it's also behind the mechanism of how you end up with blood transfusion rejections and organ transplant rejections. You end up with the body's immune system seeing these cell recognition proteins and coming to the conclusion these cells are not supposed to be here, so we need to get rid of them. So junction proteins, we said, basically either give a cell structural stability and rigidity, or they provide some special type of physiological function. In the case of gap junction proteins, which you can see one of them right here, these are multimeric proteins that span across the membranes of two different cells that are right next to each other, and essentially creates a tunnel from the inside of one cell to another. The idea of how your heart beats, and if you want to learn more about this, you could maybe take my human physiology class another time. The way this works is that the pacemaker, which is called the sinoatrial node of the heart, produces excitatory molecules that are going to spread throughout the cardiac muscle of the heart, and these excitatory molecules can move from cell to cell through these open gap junctions. This allows all the different cells that make up the cardiac muscle of the heart to all get excited at the same time so that they can contract and relax as individual units, and we can get an effective mechanism for pumping blood throughout the body. And then finally, if we want to look at an example of a receptor protein, a good place to start would be the insulin receptor, which the insulin receptor and insulin itself, you might be aware, is central to the idea of how people get diabetes. So insulin and the insulin receptor function in allowing glucose, once it is absorbed into the bloodstream, to enter in cells to where it can be used. The problem is that glucose is a very polar molecule, and polar molecules cannot get across the plasma membrane of cells without a transporter protein, like we were talking about before. However, if insulin, the hormone insulin, not the protein, or not the receptor, but the hormone insulin, which actually is a protein itself, if the hormone insulin is not here and bound to its receptor, then glucose cannot get in, because these transport proteins for glucose, which are called GLUT4, they are stuck inside the cell in what you might call a holding pattern. They can't do their job if they're not up here at the plasma membrane to let glucose in. However, when insulin does bind to the insulin receptor, this is going to precipitate a cascade of signaling events inside the cell that basically is going to result in these GLUT4 channels being inserted into the plasma membrane, so they can now do their job and allow that glucose, which is out here in the bloodstream, to enter into the cell where it can be used for creating energy that the cell needs. Okay, thank you so much for sticking around until the very end here. I know this was kind of a long set of videos, but we are finally done here. Definitely, if you have any questions about the material that's presented here, if we were living in more normal times where I could actually lecture on this face-to-face, I would be able to field questions as we go throughout here. So definitely, as you kind of reflect on the content of these videos, try to come up with questions, write them down if you feel like you're not going to remember them, bring them the next time you come to class, and I will do my best to answer those questions. I definitely want to make sure you get as much out of this class as humanly possible, and you're not going to get as much as you should out of this class if you have lingering questions that never get resolved, and I want to do my best to resolve those questions. Okay, so thank you again for your attention, and I will see you next time for your next virtual lecture. So long.